Section I. Study of carbohydrates, fats, and proteins in pure form and detection of their presence in given food stuffs. 9.1 Experiment. Aim to study carbohydrates, fats, and proteins in pure samples and their detection in given food stuffs. Requirements: test tube stand, glass holder, Bunsen burner, etc. Chemicals: Fehling's solution, Benedict's solution, AgNO3, NH4OH, NaOH, CuSO4, millions reagent, chloroform, potassium bisulfate, benzene, HNO3, etc. Theory: cell is the basic structural and functional unit of living organism. It contains 70% of water and remaining is organic matter. Carbohydrates, proteins and fats which are needed for the growth and development of cells. Body obtains these nutrients from food we take. Carbohydrates. These are polydroxyaldehydes or polyhydroxyketones or compounds which yield these compounds upon hydrolysis. Sugars and non-sugars. All the monosaccharides like glucose, fructose, etc. and disaccharides, sucrose, maltose, lactose, etc. are sweet in taste. They are called as sugars. Polysaccharides like starch, cellulose, etc. are not sweet in taste and are called non-sugars. Reducing and non-reducing sugars. Those carbohydrates which can reduce Fehling's solution or Torrance reagent are called reducing sugars. Which do not reduce these reagents are called non-reducing sugars. Proteins. These are the condensed polymer chains of proportional to amino acids. Condensation of negative NH2 group of one proportional to amino acid with negative COOH group of the other proportional to amino acid with the elimination of a molecule of water results in formation of a dipeptide, and the CONH bond formed is called a peptide bond. When a large number of proportional to amino acids are joined by peptide bonds, the polypeptides are formed. Polypeptides having molecular mass greater than 10,000 daltons are called as proteins. Oils and fats. These are triesters of glycerol with unbranched higher fatty acids. Those which are solids at room temperature are called fats, and which are liquid at room temperature are called oils. Oils contain higher proportion of unsaturated fatty acids, while fats contain higher proportion of saturated fatty acids. Test for carbohydrates. Test. Observations. Inference. Fehling's test. Take 2 ml of sample solution in the test tube. Add to it 2 ml of Fehling's solution, 1 ml Fehling A and 1 ml of Fehling B. Heat the test tube on a water bath. Red precipitate, carbohydrates present. Benedict's solution test. Take 2 ml of sample solution in a test tube and add 2 ml of Benedict's reagent. Heat the test tube for some time. Red precipitate, carbohydrates present. Molish test. Take 2 ml of aqueous solution of compound in a test tube. Add 2-3 drops of 1% naphthol. Now add concentration H2SO4 along the sides of the test tube. A reddish purple ring is formed at the junction of two layers. Carbohydrates present. Tolerance test. Take 2 ml of sample solution in a test tube and add 2 ml of AgNO3 solution and 1-2 drops of NaOH solution. Then add NH4OH dropwise with shaking. Heat the test tube on a water bath. Formation of shiny layer of silver along the sides of the test tube. Carbohydrates present. Iodine test. Test for starch. To a little sample, add a few drops of iodine solution, deep blue color, carbohydrates, starch only present. Test for proteins. Biuret test. Take a little given substance in a test tube, add a few drops of 10% NaOH and a very dilute solution of CuSO4, appearance of violet color, protein present. Nenhydrin test. Take a little given substance, add a few drops of 0.1% nenhydrin solution, boil the contents for some time. Blue color on cooling, protein present. Xanthoproteic test. Take a little given substance in a test tube, add 5 ml of concentration HNO3, shake it and leave it undisturbed for some time. Appearance of yellow or deep yellow color, protein present. Millions test. Take a little given substance in a test tube, add about 2 ml of Millions reagent and heat the mixture, white PPT, which turns red on heating, protein present. Test for fats. Solubility test. Take 5 ml of the given substance in a test tube and add chloroform or benzene. Substance dissolves, fat present. Acrolein test. Take 5 ml of fat or oil in a test tube, add 2 to 3 drops of potassium bisulfate, heat the test tube gently at first and then strongly, irritating smell, oil or fat present. Spot test. Pull a small amount of the given substance on a filter paper, fold the paper and press it tightly. Appearance of translucent spot on unfolding the paper, oil or fat present.